that after 2 years I would be good at this.
Right.
All right, is everything clear?
Perfect.
Okay, so I'm going to talk a little bit about preliminary results that I've got over the years. This work has been severely delayed, unfortunately, by the pandemic, but I hope to finish this in the next year or so. So echinoderms, they are marine organisms, and they're currently represented by 5 living classes. The fossil record is huge, but only 5 living classes made it. And the class Echinoidea is composed by sea urchins, hard urchins, sand dollars, and the cassiduloids. So the class Echinoidea originated about 450 million years ago in the Paleozoic. And the first sea urchins, first echinoids, I should say, they're all globose and with big spines. Everything you think of a sea urchin today, that's how the first echinoids look like. And there were several lineages of sea urchins back then. And then we had a major extinction at the end of the Permian. It's called the Permo-Triassic mass extinction event. And that decimated a lot of marine organisms. Also on land, it was drastic, but it was very severe in the oceans. And this extinction was triggered mainly by intense volcanism. That's a major driver of that extinction and all of the consequences that happened afterwards for millions of years. In the case of the echinoids, many lineages were extinct, as I mentioned. Just a few survived, but only one really made it to the present. So all of the others that survived, they went extinct just a few million years after the extinction. And then during that period that followed the extinction is around the Mesozoic era. And we call that time the Mesozoic Marine Revolution. This is a very exciting time, like following an extinction, when we eliminate a lot of species, that you empty a lot of ecological space. Then you have a revolution that we see in the oceans of marine organisms, that we have the predators, for example, the crabs, the ichthyosaurs, the marine snails, they were finding better and better ways to prey. And then we have the prey finding better and better ways to evade predation. So we have this evolutionary arms race between predator and prey. It was very exciting in the ocean, and that drives the diversification of many marine organisms. In terms of the echinoids, we can see that the groups, they started burrowing in the sediment, what we call infaunalization. And in that time, that drove a lot of morphological change, like that pressure to evade predation, and then drove the diversification of a clade of echinoids that we call the irregular urchins. So the irregular urchins are this group in the green shaded area. And then on the other side, we call the regular urchins. They're not really a monophyletic group, but we call them regular because when you think of a sea urchin, you think of something like that, globose and spine. And here the irregular urchins, you're seeing them without the spines, but they do have spines, just because generally when they find them in museums, when they're dried, the spines have fallen off, but they are the sea urchins. And just to give you a sense of time, when the echinoids were doing the revolution in the water, is around the same time that the dinos were doing the revolution on land. And then after that period, and as we all know, at the end of the Mesozoic area, we have another mass extinction event, which is called the end Cretaceous mass extinction, that was triggered by a meteorite impact, but also we have records of volcanism, not as intense as in the Permian, but also there was some intense volcanism in the time. And then this extinction was more severe on land. And then what we see in the oceans, that even though the diversity of echinoids decreased, we don't lose any major lineages. So different from the other extinction, this one, everybody made through, we just changed a little bit the dynamics, diversification dynamics. So we have 2 major clades of irregular echinoids. So we have in the blue shaded area is the neonatostomata, which is composed of the cassiduloids and the sand dollars. And then we have the atellostomata, which are the hard urchins. And if we plot the diversification through time of these two groups, we can see here in the y-axis is the general diversity, and then the x-axis is the time. So going to the right to the present, we can see that both groups are diversifying over time, even though the hydrogens are more diverse than the cassiduloids and clypeasteroids and centauroids, sorry. Both are diversifying through time. However, if we break it down into one group, which is just the neonatostomata, we can see that, sorry, there's a helicopter over here in the middle of DC. So if we break it down, the neonatostomata, we can see that the diversification of the group is mainly driven by the sand dollars. And the cassiduloids, which is the group below, even though they diversified in the beginning of their existence, they started declining after that. And today they're very rare in modern oceans. So the cassiduloid fossil record is extremely rich. There are way more fossil specimens in museums than there are living specimens. About 40% of the whole class diversity in the Eocene, which is a little bit after the end Cretaceous mass extinction, 40% of all echinoids was composed of cassiduloids. And then they drastically declined after that, and today there are only about 32 to 35 living species. They showed a very interesting reproductive strategies. So we can see here oops, let me look for my pointer. This specimen here, if you can notice in the middle here, there is a shaded yellow area. So all of these, I counted about 200 baby urchins that were on top of on the body. So you can see their larvae, they're held between the spines, and you can also see young juveniles that look for protection among the spines of the mom. And then you can see also an extreme brooding that actually involves a brooding chamber, which is in these species here. And here, if you can see, this is a micro CT scan of a Tropholampas lovening species. And you can see here in the middle, there is this darker area, which is actually a brooding chamber. And I found 3 babies brooding inside the spines in that chamber. So they have varied strategies, not only these two, but these are the main, most different ones if you compare it to other cassiduloids, other urchins, sorry. And they also display a very interesting geographic distribution. So you can see here, all of these spots are different species, and the colors refer to families. So they have very restricted geographic range. They live, except for these species here in the Pacific that has a broader range. They generally live in a very small area, and most of them are tropical or subtropical. A few are subtropical, and it's very spotty. If we plot here the fossil record, you can see that the distribution of the pests is way more widespread, and we can even find some species down here in Antarctica and further north in Canada. So places that today are cold, but then we have to see how the temperature was back in the day that they lived there. They have very interesting distribution. I've been wondering what drove this pattern through time. So there are 2 hypotheses for their demise, for the cassiduloid demise over time. Sorry, there are many hypotheses. I'm going to talk about 2 hypotheses because they relate more to the results that I'm going to show you later. One is Cenozoic cooling. So you can see here in the right side, there's this graph that shows that in the last 65 million years, so this going from the past to the present, we can see that we were living in a hothouse. And then at some point, about 40 million years ago, we started cooling down. And today we live in a cold house, even though currently we're driving the temperature to the roof. We are technically in the cold house if you take into consideration 60 million years in Earth life. And here in the top is a reconstruction of the map of the globe. So you can see here that in the hothouse, the sea levels were really high. There were no ice in the poles. So many of the continents were flooded, and that makes it possible for many species that like to live in shallow water. All of these lighter blue area There are shallow oceans that species love to live and diversify. So there are plenty of them. Here was the Tethys Ocean. And you can see that after the cold house, the oceans were drained. There way less area for shallow water species to live. So there were a major change. And one of the hypotheses for the cassidyloid decline is this change. Another one is morphological constraints. So you can see here on the left, there are living species. So all of these belong to different families, and you can see that they're very similar to each other. It's very hard to come up with characters to reconstruct the phylogeny of the cassidyloids because of this. They're very similar. It's very hard to come up with difference other than shape and number of pores, for example. So I'd like to bring your attention to these structures here. They're called the petals. They have modified breathing structures for all the urchins. They have these structures, these pores here, but in regular urchins, they breathe on the surface using these petals. And you can see that all of them are kind of similar. They're all flush. They only change in shape and the number of pores. But if you compare here with the hard urchin, you can see that some species evolve these funnels in the petals, and that allows them to make tunnels within the sediment such that they can breathe the water in the surface. So they can burrow very deep in the sediment and keep breathing the water in the surface. That allows them not only to burrow deeper in the sediment, but also to live in finer sediment. So they can live in mud, for example. Cassidyloids, on the other hand, they don't have the ability to do that. So they can only burrow a few centimeters below the sediment. And also they can only live in coarse sediment. Oops. Okay, so you can only live in coarse sediments because they need the water within the grains to be able to carry oxygen around. So they're very restricted in terms of habitat with regards to the hard urchins and the which are other irregular echinoids. Also, there is some limitation in terms of burrowing. Hard urchins, they have many different types of spines that help them burrow, while cassidyloids only have one type of spine. So this is a time lapse of a hard urchin and a cassidyloid burrowing. And you can see that 1 minute and a half later, the hard urchins are almost completely gone. 4 minutes later, the cassidyloid is still trying to burrow. And they start at the same time. So you can see that the cassidyloid is much slower. And if there is a marine snail around, which can eat both of them by drilling holes on the shell, I can see here. If there is a marine snail around, the cassidyloid will most likely be the prey, and the hard urchin is going to evade predation. So cassidyloids haven't done much to, as the hard urchins have, to drive that diversification either. So I started investigating the timing of the diversity decline and then the geography of the decline by doing the cassidyloid phylogeny. So here I collected data from the skeleton. So you can see here, we call the plate architecture. Echinoids are made of many plates. They are bound together by a connective tissue. So the shape of these plates, the organization of these plates are important. So I collected data on these. I collected data on accessory structures that we call pedicellaria. I made several measurements. And then I used that to reconstruct the phylogeny. And just call out your attention for, I mentioned that it's very hard to come up with characters for cassidyloids. And it was very important for me to do a micro CT scanning of the living species. I'd love to do other fossil ones too, but it hasn't worked yet because they eat sediment and there is not enough contrast between the sediment and the skeleton to see the structures inside, but it's worked really well with living species. So here I did this study at the Berkeley lab. They have a synchrotron. The specimens were about 2 centimeters, just the perfect size to put in the X-ray hutch and get amazing images. So you can see here, these species are only 2 specimens in the world, the holotype and the paratype. So I would never be able to break them, but using micro CT, I was able to scan and see perfectly inside all of the structures in the test. So this is very important for my work. So here is the cassidyloid phylogeny. I'm not going to focus much on these. This has been published a couple of years ago with collaborators. I just want to show you here the diversity decline, the timing of the decline with the temperature change. This is the first test of how effective the temperature could have been to drive the diversity decline of cassidyloids. So you can see here these, the bars are the stratigraphic ranges of the species. So basically when the species lived, and in the bottom we have the geologic time scale. So you can see here, this red bar is showing when the hothouse was, and you can see that there were many actually was when the cassidyloids were diversifying the most during the hothouse. And then we can see the moment later when it started cooling down, most of the lineages went extinct at that time. So you can see that indeed it makes sense to investigate the hypothesis because most of the lineage went extinct with the onset of global cooling. So next, I was investigating the process driving the geographic distribution of the group and see, for example, those species in Antarctica, they were there when it was cold or when it was hot. If they like the hot, they shouldn't be there. So I used a software called RASP with this preliminary data to reconstruct ancestral states. And I used occurrences from the paleobiology database, a huge database with fossil occurrences. I used models for global circulation and also the changing configuration of the continents because of plate tectonics. So based on the occurrences of the fossil and recent taxa, I defined 8 geographic areas, which are these areas here where cassidyloids live. I should mention that I focus on only 2 families of cassidyloids. Next, I'm gonna move on to the other 2 families that wasn't included here. So first, I use a DEC model using 2 Q matrices, one for range constraints. For example, in this case here, I assume that to live in South Tetis and North Atlantic, which is here, would be a probability of about 1. But for a species to live only in the East Pacific and in Madagascar was a probability closer to 0. So this was one of the Q matrices, and then I also infer probabilities for dispersal constraints. And in this case here, the model allowed me to, the software allowed me to do a time stratified dispersal constraint matrix. And that's because of, again, the changing configurations of the continents and also the current configuration. So you can see here that before 35 million years ago, during the hothouse, we can see that there was a global equatorial current going from east to west in the middle of the continent. So that was a major circulation in the globe, in the oceans. And you can see that after that, Africa and India, they moved northward, and that caused the formation of the Mediterranean Sea. It caused the formation of the Himalayas, but also it blocked that current that was going in the middle of the globe. And it started a new system, that's the system that we have today, that is the system of gyres. So today we have gyres that are confined to the oceans. They don't cross again from one to the other in the middle. So it's a very change in configuration for the currents in the oceans, and that completely changed the dispersal patterns of the species. And another event was the closure of the Isthmus of Panama. So we have here northward movement of America and also the build of this land bridge, the Isthmus of Panama that caused Vicarians. So before we have species that lived both in the Caribbean Sea in the East Pacific, and now after the Isthmus of Panama, that passage was mostly closed. So I also took that into consideration to build this matrix. So how far again, I focused on 2 cassidyloid families, and these are the results for those 2 families. You can see here, again, the bars are the stratigraphic ranges of each species. You can see the geologic time scale on the left, and the colors refer to the colors on the map. This is about Miocene, about 35 million years ago configuration, these maps. And you can see there's some instances of long range dispersal in the graph on the left, which was a little bit interesting for me because This group is the one that have most of the brooding species. So I wouldn't expect long dispersal necessarily, but it recovered long dispersal for that group for a couple of species. We can see here the shaded area is the reconstructed ancestral range, is the same for both families. And there are three main takeaways that we can take two patterns coming from those two configurations, those two events. So first, the center of origin was a major exporter of taxa. So actually you can see is range extension of the cassiduloids instead of, I mean, they have such a spotted distribution today, but at some point in the past, they actually went around a lot. And the dispersal was mainly eastward, which is against that global current that I mentioned before. The current was going from east to west, with a lot of dispersion going on the other way, and that was before the closure of the Tethys Ocean and the formation of the Mediterranean. So that was interesting. And also there was 2 more times dispersal than Vicarian events. We generally expect that for the oceans because it's a free, almost free for all for certain groups that can disperse very easily. So there was more dispersal events and not many area extinction events for these 2 groups. So next, I also have been investigating the evolution of body shape to see how constrained the morphology is. So I collected geometric morphometrics data on the test outline. So test is the echinoid body. So the outline of the body, I got landmarks and semi-landmarks. And then I also got details of the petal outlines, the petal of those breathing structures. So I'm showing here the whole test, but I actually did only with half of the test because it's nearly symmetric. And I used the geomorph package in R to do the analysis. Just to show you this plot here, the PCA plot, completely mixed up. You can see there's no difference between those groups. I'm going to focus on each separate one later based on time, but I just wanted to show you that within the groups, among the groups, there was no difference in body shape. But if we divide them by time, we can see a very interesting pattern. So here I'm focusing on the end Cretaceous extinction. So I mentioned that didn't change, they didn't eliminate any of the major lineages, but it changed diversity within them. So here I'm testing for, did it also change the shape for those cassiduloids? So you can see here, the grayed out are Mesozoic species. And then we have in yellow, in blue, the ones that were in the late Cretaceous, which is before the extinction. And then in yellow are the ones right after the extinction. So we can see that before the extinction, the morphospace was occupied by all of these species here. So this is basically a PCA plot. Sorry, I should have said that. A PCA, principal component analysis plot. So you can see here the blue dots are the shape before, these with respect to the outline. And then after the extinction, we can see that all of this area here was mostly vanished from the cassiduloid morphospace. And then we can, just to see what exactly those shapes mean, these groups on this side here and up here they have a more elongated shape and a very funneled posterior region. And these species living here, they're mostly a round shape. And we assume that having an elongated shape help you to burrow faster. The hard urchins, they have elongated shapes. And most of the burrowing cassiduloids, they have elongated shapes. So you can see an extinction of these morphologies here. And then if you move forward to the next period, we can see that this space was sort of reoccupied. So the orange is the recent, there was a recent, after the Eocene, all of the species, and this is the reoccupation of that space. But we can see which species are reoccupying that space, and we can, they are round shape, but they're not burrowing. They're very big. They generally live in the deeper waters. And we can see that they're actually not burrowing. They're protecting themselves in some other way. And then when we turn to the petals, we see a different story. So here I plotted all of them together just to show that here in the late Cretaceous to the next stage, there wasn't much difference in terms of extinction. So extinction did not remove. Actually, it expanded after that. So we can see that not only right after the extinction, but towards the present, we have been, the aciduloids have been changing a lot their breathing structures, not to the point of the hydrogens to provide like new innovations to build tunnels and etc. We don't know exactly what those differences mean, but there has been some differences after the extinction event. So I'd like to draw some conclusions here from these very preliminary results. I'm still working on all of these in different ways. So we can see that there was onset of the diversity decline in the aciduloid, which coincided with global cooling. I still want to improve the calibration of that phylogeny because I just use the stratigraphic ranges in here, and I want to include uncertainty of the fossil record. But I would say that it pretty much follows the story as it happened, as you can see from the fossil record. There's been dispersal to new areas. There was a major driver of speciation in aciduloids, which is something, again, that I did not expect much, even though, yes, I expect them to diversify, to disperse and diversify because they are marine organisms, but at the same time, there are a lot of brooders. But that being said, I don't know exactly what happens in the fossil record because we don't have, it's hard to tell what the fossils did because we don't have the babies within spines and no spines left to see that. And then we can see extinction selectivity across the interpretation extinction in terms of shape, body shape, against a round body shape, again, that we assume that is worse to burrow. But in terms of petal shape, it diversified, and that could be a proxy for improved breathing. And with that, I'd like to thank Christiane, Dolores, and the Willy Hennig Society for hosting me, and all of you for your presence. Thank my collaborators, funding agencies, all the museums, and other institutions that supported this work. Thank you very much, and I'll take your questions.